Welcome to the webinar, everyone. Thank you for coming. Welcome to the October Fireside Zoom Chat for Leadership and Community-Driven Connections to Research with citizen scientist Marian Lemle and ME CFS Researcher Bindu Paul. Good morning, everyone. This meeting will be hosted today by Ovid Amate, the President and CEO of Solve ME. Good morning, Ovid. How are you?
Good morning, everyone, and good afternoon for those of you who are on the East Coast. And really, thank you so much for joining us for this very special webinar that we're having today. And it seems that these days it is very important for all of us to highlight the importance of science. So today we are not only celebrating science, but also recognizing a journey that is as much scientific pursuit as it is a personal mission. And you'll hear about that in a moment. We're celebrating what an individual, in this case, a citizen scientist, can do to advance our understanding of a devastating disease and hopefully where it can take us from there. But before we do that, I just wanted to provide a very brief update on the here and now. This is an opportunity for us to connect. So I didn't want to miss that. And of course, at this particular moment in time, we're in a world where politics and the COVID pandemic, of course, are part of our daily lives and in every conversation. So to start, I just wanted to share a quick update on the hard work that we're all doing to help pass the House Resolution 7057, which, as you may know, was introduced by Representative Jamie Raskin. And this is the House resolution that calls for the Understanding COVID-19 and ME CFS subsets, which will be the first piece of legislation to authorize funding from the NIH for ME CFS research and education. And at this point, we have nearly 50 supporters, nearly 50 representatives with a bipartisan support for this legislation. And so I'm not going to venture and predict the political outcomes at this stage, but we're hoping that we can still make a difference, even in the uncertain political climate right now. To make sure that we can continue to move forward with that, perhaps in a lame duck session. So we continue to push for that, and of course, all of your support and help makes tremendous difference. But I also wanted to remind us that during this year we were able to prevent the elimination of the Centers for Disease Control budget. So the CDC still has a program that preserved more than $20 million in funding for research and education. And of course, the recent news that there were several grants that received more than $500,000 in grants as a result of the work that opened defense research funding for ME CFS. So these are all very important aspects of how making sure that the political involvement gets the support for our community that we so badly need. On a different front, I wanted to update you on where we are with the You and Me registry, which, as you know, was recently launched, and we are already at more than 1,700 people registered. The majority are ME CFS patients, but we also have a growing number of people who are controls. So that's really remarkable, and I hope that over the next several months we can begin to share some of the initial insights from this growing body of data that we're all collecting together. Also wanted to mention that we're obviously recognizing the need to study people who continue to struggle beyond their initial infection with COVID-19 in what seems to be very similar to the ME CFS experience. And so we will be opening the registry so that we can actually document and collect the data from people who are currently not diagnosed with ME/CFS but continue to experience symptoms after COVID-19. So pre-registration for that is already open, and testing of that part of the registry is already ongoing by members of the community, and we're looking to open up more broadly next week. So these are all things that are happening in real time, and I just wanted to provide you an update on that. And now for today's topic. So as I mentioned, this is really a story of science, but really a personal scientific quest that got started long before I joined Solve. So this is really an opportunity for me to introduce Carol Head, that I don't think really needs an introduction to this forum, but gives me an opportunity, Carol, to thank you for your leadership and your contribution, and to make this particular story that we're sharing today for you to help make it real. Carol, please take it away.
Thanks so much, Obed, and I'm delighted to be here and support you in your role as CEO. I've been tasked this morning with introducing two researchers. First, Marian Lemle, and then Dr. Bindu Paul, who are now working together. So let me talk a little bit first about Marian. So she is a creative thinker and a citizen scientist. You know, like so many, when her daughter became suddenly ill with ME/CFS, she worked diligently to comprehend the underlying causes, and she began a systematic study of the literature and developed some initial ideas. Chance favors the prepared mind. So while attending a lecture on induced hibernation in mice at a meeting of White House Fellows in 2006, where, by the way, she had previously given a talk about creative thinking, Marian herself had a spark of inspiration, and that was this, that people with ME/CFS perhaps didn't need an injection of hydrogen sulfide gas, the focus of the mice model study. Perhaps they were already in a dialed down state resembling hibernation, and that this gas, hydrogen sulfide, possibly had something to do with ME at a molecular level. She theorized that the gas was possibly affecting the body's ability to fully utilize oxygen. So that insight led to the development of the hibernation theory of illness in ME/CFS and represented the first ever comprehensive theory of hydrogen sulfide in chronic disease. Marian then produced a paper in 2007 and published it in print ahead of its publication in September 2008 and testified before CFSAC of that same year. She also presented a poster on her theory at the IACF SME conference in 2009. Several key aspects of her ideas concerning hydrogen sulfide and the bioenergetics of the mitochondria have since been confirmed. In August 2016, a second study of the metabolomics profile of ME/CFS patients provided additional support for the theory that ME/CFS patients may be experiencing hypo-metabolic state, akin to hibernation. Marian then became a project development consultant at the Brain Research and Integrated Nutrition Center at Nova Southeastern University, where she advocated for research on hydrogen sulfide and participated in a research project at B12 and hydrogen sulfide metabolism. So just to back up a little bit about Marian, because I find this so intriguing. Earlier in her career, Marian had worked in policy planning and development at the White House Office of Telecommunications Policy. She's also served on 4 nonprofit educational boards. She holds an MBA from the Wharton School at the University of Pennsylvania, where she was a noted teaching assistant in venture initiation and entrepreneurial development. She received a certificate in painting and drawing from the Corcoran College of Art and Design, where she was awarded the Lester Prize for painting. And if all that weren't enough, Marian also recently descended over 2,000 feet below sea level in a small submarine to witness firsthand life forms that thrive in an environment of diminished oxygen and total darkness in pursuit of her scientific theory. So I'm introducing two researchers today, but please note that Marian is here today and brings zero formal education in the sciences, and yet has developed this theory, which is becoming more and more impressive and regarded as critical. It's a highly compelling research theory. So when I think of Marian, I have to say the words Renaissance woman do come to mind. I also have the pleasure of introducing Dr. Bindu Paul, who grew up in India from a young age traveled all over the country. During her frequent moves, she was exposed to different climates, cultures, and people, and quickly learned to adjust to different conditions. So even as a child, she loved science and was always experimenting at school. She obtained her PhD from the Indian Institute of Science in Bangalore, where her professor introduced her to the fascinating world of DNA binding proteins and transcription. After postdoctoral work at the NIH, our Dr. Paul joined the laboratory at Johns Hopkins University, where she focused on antioxidant defense pathways mechanisms effective in neurodegenerative diseases. She discovered a depletion of cystathionine gamma lyase in Huntington's disease, which mediates disease progression. Dr. Paul recently examined the immune cells from a small cohort of patients and healthy controls from our own Solve CFS biobank. Her preliminary findings are incredibly compelling and opened up several lines of investigation and collaborative research, which she continues to pursue. We're thrilled that such an exciting early stage investigator is leading research into ME/CFS as part of our Solve ME research program, and we are providing funding in support of her work. So with those introductions to extraordinary women, Marian, I want to turn to you with a question, and just briefly, how did you get started in this journey of exploring ME/CFS?
Well, Carol, first of all, I have to say thank you for that extraordinary introduction, and Ovid, thank you. And thank you for having us here today. It's really an honor. As you know, and as you mentioned, most people are brought into the world of ME/CFS because of an illness in the family. And in our case, it was our daughter who came down with an unspecified virus in her senior year of high school. She was well enough to go off to college, but in her sophomore year, she became very ill again. This time, incredibly ill, and she had to drop out of college. She came home and she was bedridden. At that time, chronic fatigue syndrome had the name the yuppie flu, and it was very difficult to find doctors who were willing to take it seriously. They all deserve such a shout out, believe me. And there just weren't answers. So I started researching everything I could find, particularly from PubMed and on the internet. And I happened, as you mentioned, to attend this lecture, the subject of which was, when does life really end? And the researcher was working for the Defense Department, trying to figure out a way to stabilize soldiers on the battlefield so that they could be transported in a state of suspended hibernation to a hospital where they could then be sort of brought back to life to be helped. And as he was describing what happened to these mice after he had pumped hydrogen sulfide, a toxic gas, into their tank, they became essentially half dead. And I said to myself, my gosh, that sounds like my daughter's situation. And so I then started researching this. So could we have the first slide? It turns out that in 1977, there was a discovery of deep sea vents from which all sorts of gases were emanating. But next slide, please. but one gas in particular was of interest, hydrogen sulfide. There are many scientists who believe that the deep sea vents is actually where life began. In any case, surrounding these vents in total darkness and without oxygen are the 15-foot sea tubes, and they do not need oxygen or light. It turns out that as we evolved onto land and breathed oxygen, our need for oxygen became greater, but our body still retained the hydrogen sulfide. And so I was wondering, well, does this possibly mean that we have too much of that in our bodies, or as we've since learned in the last 15 years, too little? Next slide. At around the time I was beginning my research, and I was helped in this by a family friend, Dr. Carl Peck, who was at the time the Assistant Surgeon General of the United States. This paper came out, photosynthesis, but chemosynthesis, and it turns out that hydrogen sulfide could be used by the mitochondria as an essentially a backup system for energy production under stressful conditions. Next slide, please. So the question was, could our cells actually be breathing this largely toxic gas? Next. So I, with Dr. Peck's help, I put together a paper. Next slide, please. And here are some of the key concepts. I don't want to spend too much time on this because the science was there in my paper, but what's really impressive is where it's going and where it has gone since this time. But in any case, so I highlighted metabolic hypo-function, homeostatic imbalance that went to the molecular level. And it was clear that you always had to think about chronic fatigue within the environmental context and the context of one's genes, particularly infections, whether they were bacterial or viral. And it did seem that this could provide a sort of unifying theory. In terms of the mitochondria, hydrogen sulfide and oxygen were imbalanced in ME/CFS, and hypoxia, that is the body's ability to utilize oxygen might be compromised. It was clear at that time, even at that time, that redox, reactive oxygen species, and ATP production were key. Next slide, yeah. I mentioned that there was possibly a protective mechanism working through the iron sulfur clusters, cytochrome C oxidase at level 4 of the electron transport chain. That's very specific, and it's interesting. A paper came out last year that said that Down syndrome actually can be traced to an overexpression of one of the enzymes, cystathionine beta synthase, CBS, at level 4 of the electron transport chain. And what's so exciting about that is that having pinned that down, there actually may be some therapeutics. So I had some other thoughts about how it could trigger a chain reaction in the mitochondria. And also it's very important that the bacteria in our gut basically retain this oxygen and hydrogen sulfide balance. And when that gets out of balance, you can have all sorts of problems. And also, this will figure in Bindu's research, during an inflammatory response, there are changes in sulfur metabolism, and the mitochondria actually hold on to it, particularly glutathione and cysteine, and our bodies need more of it. So I thought this was interesting. This is an old slide, but these things are still being talked about today. The blood lactate concentration and lactate pyruvate concentration, anaerobic metabolism, fatty acid metabolism, the gut-brain axis, anaerobic bacteria, vagus nerve, and B12 hydroxycobalamin. These all still figure in ME/CFS. So that's the last slide. And where do we go from here, Carol?
So thank you for outlining the theory in a way we lay people can grasp it. And so just turning a little bit then, Marian, how did you first get involved with the Solve ME-CFS initiative?
Well, let me say that Solve and what was previously known as the CFS organization has been critical for me. After I wrote the first paper, I needed someone to validate it. And I spoke with Suzanne Vernon, who was then the scientific director. I think I sent it to her in 2007. And she said it made sense. It made biological sense. I then had many other experiences over the years with the other scientific directors, Zahir Nale and I always for
Sadie.
Yes, thank you. Who've all been incredibly important in supporting me and my ideas. So that's basically how it got started.
Terrific. So once there was that interest, what did you do next? What happened next for you?
Well, so what happened was that Suzanne Vernon and Kim at the time said, you really have to do something. So they encouraged me to publish. I did publish a quick one-page synopsis of this theory in the Journal of Medical Hypothesis. And also they encouraged me to make a presentation to the CFSAC committee, which I did in October of 2008. I also then produced posters and tried to interest people in the research. And it was I don't know if I should say this here, but it was very frustrating because I could see that there were enormous strides being made in the research in so many other fields, but not in ME-CFS. And that was difficult. I mean, I was lucky enough that someone at NIH, Dr. Leroy Saligan, picked up on the idea and cited my work, as have several others, but it was largely below the radar. And anyway, that's changing now as it becomes clear that hydrogen sulfide isn't just toxic, it's essential. We can't live without it. And our mitochondria actually retain that capability as we evolve to use it. So it's really actually all about balance, and too much or too little is deadly.
So, well, first, thank goodness that you persisted despite lacking formal training in the sciences. A good theory is hard to put down, basically, as the summary here. So then let's just fast forward, I think, to a couple of years ago. And since then now you're working with Dr. Bindu Paul. Can you talk a little bit about how the two of you connected?
Well, that was it's quite remarkable. And I'm so thrilled to be working with Bindu. She is wonderful. She has the most open mind. And that's an important requirement for doing good research. Essentially how this evolved was that Carol, actually, you and Zahir Nale sought me out and said, why don't you get back into this? Your theories are so important. And we feel you have a contribution to make. So I updated my paper, and it was sent off to Dr. Tony Komarov, one of the world's leading experts, to validate. And he said, you're absolutely on the right track. And during that conversation, he asked me who my dream team was. And he said, whose papers really get you excited? And I said, well, there's someone at Johns Hopkins, Dr. Solomon Snyder and someone named B. Paul. I didn't know B. Paul was a woman or a man. And he encouraged me to get in touch, but I was intimidated. Anyway, long story short, someone I know did put me in touch with Dr. Oh, I'm sorry, I'm getting so nervous about this. With Dr. Solomon, he likes to be called Sol, and I had a conversation with him. It was quite nervous making because in half an hour he said only a couple of things, and afterwards, when I spoke to the friend who'd made the introduction, I thought it had gone very poorly. And she said, oh, no. He thought you were great. He was very impressed. And so with that, as you know, I then worked with Sadie to see if we could invite Bindu Paul, who I learned was a woman, and Dr. Snyder to the NIH conference. And I was so thrilled when Bindu showed up and we started talking, and we haven't stopped since.
You know, it's fun to hear this because this is how science really happens, right? These connections. So Dr. Paul, can you talk a little bit about why you were intrigued by this and interested in talking with Marian?
Yeah, sure. First of all, let me thank you for inviting me to this meeting. It's really an honor and pleasure to be here. Yeah, now regarding how I got involved with ME/CFS. So one morning when I logged into my email, I saw a message from Sadie Whittaker and Alison Ramiller from Solve ME, inviting me to a conference which was to be held in April of 2019 at the NIH in Bethesda. And they mentioned that they really liked our research on redox signaling and hydrogen sulfide and cysteine and glutathione and all of that. And maybe that could be applied to ME/CFS. And if something nice came out of it, it would benefit millions of people across the globe who suffer from ME/CFS. So, the metabolism of H2S and cysteine has been very interesting to me for a long time. I've been working with Sol for the last 15 years. And believe it or not, among the conditions which have low cysteine, ME/CFS was on my list, but I never had the resources to look at cysteine metabolism in HD. In ME/CFS. I was mostly working on Huntington's and Alzheimer's and Parkinson's. And then I thought, okay, why not? Let me just attend the meeting and hear about the research in ME/CFS. And so it was a short trip from Baltimore to Bethesda. So I went to the conference and then I met all of the people there and I was really impressed. And then I met Marian, of course. And I was very struck by how passionate she was about the science behind the chronic fatigue symptoms and how involved she was. And she really knew a lot. And that impressed me very much because it's very rare that, somebody from a non-scientist background really goes into such depth. And then, of course, we got along really well, and then we discuss a lot. And like Marian said, we continue to discuss, every other day. And that's how I got into ME/CFS.
Well, and we're delighted that you did. Could either of you then, before I turn it over to Ovid to take questions, can you talk just a little bit about the status of your work together today?
Okay, so to begin with, I'll just introduce, our research on hydrogen sulfide, which has been going on in Johns Hopkins. And believe it or not, all the principles that I apply to neuroscience also apply to ME/CFS because the brain is also affected in ME/CFS because you have cognitive dysfunction and the brain fog, as you name it. But one of my, I would say my favorite discovery in the world of neuroscience is the depletion of cystathionine gamma lyase, the enzyme which makes cysteine and hydrogen sulfide in Huntington's disease. Now, Huntington's disease is a neurodegenerative disease which affects the striatum of the brain, and it's caused by a mutation in a single gene called Huntington. And this leads to depletion of cysteine and hydrogen sulfide. And this publication appeared in Nature in 2014. Next slide, please. So hydrogen sulfide was known as an environmental toxin. And of course, it has got a very noxious odor. It smells like rotten eggs. And high doses of it, as Marian mentioned, is deleterious. It affects the mitochondria and shuts it down, basically. But what is interesting is that this toxic gas is also produced endogenously in the body and at very low levels, but which supports mitochondrial function, and it participates in diverse physiological processes such as response to inflammation, neuroprotection, maintenance of blood pressure, and also certain organellar stress responses. So if you're interested, you can go to the details of the science behind it in the papers listed here. For the future, we plan to look at H2S metabolism in ME/CFS, and I was fortunate to receive samples from the registry at Solve ME, and then we have some very exciting preliminary results which show that hydrogen sulfide metabolism is dysregulated in ME/CFS, and we plan to expand on that. In addition, Solve ME also put us in touch with Paul Fisher and Sarah Nestle at La Trobe University in Australia, and they have some immortalized cell lines from ME/CFS patients, both mild form, and severe form, the cell lines. And we plan to use these cell lines to look at mitochondrial function. So that's been going on as well. Yeah.
Well, terrific. Thank you. And we all eagerly await the results of the next steps of your work. It seems so promising, and partially because it addresses the very foundational mitochondrial issues potentially for ME/CFS. So
exactly,
we're excited to learn the next steps on that. So I think at this point, unless Bindu and Marian, either of you have some other comments, I know there are people with questions for you. So I'll turn this back to you, Obed, to take some questions.
Thank you so much. This was incredibly interesting, and as I said, it's a journey that starts with a personal and professional ideas, and we just begin to see where this could really lead us. And personally, it's always intriguing to see how ideas that originally or initially seem to be really coming out of left field become so logical and really begin to provide some very important clues and explanations. So thank you all for doing that. So I'll open up for more questions. I think one question that came in, and Dr. Paul, maybe it is for you, with regards to other situations. For instance, if the involvement of cyanide and its levels was looked at in the context of ME/CFS, or have you looked at that in other neurodegenerative diseases?
To answer that question, we haven't really looked at the effects of cyanide in neurodegenerative diseases, but I think that H2S could neutralize some of those effects as well. Not just of cyanide, but also of other, some metals like mercury and iron, all of that.
Yeah, that's very interesting. So, given the current situation, and of course, we realize that with the COVID situation and the restrictions on our ability to do certain things, how did that impact your research? And First of all, on a daily basis, can you actually be in the lab and continue to do experiments? How did the situation impact the study so far?
Yeah, so there are both a positive and a negative aspect of COVID-19. So let me begin with the positive. That is, COVID-19 really has given us the time to write more grants and write up our papers and apply for more funding, and which we have been partially successful in, and networking with other scientists and also meetings like this. And the negative aspect, I would say, of course, is that it slowed down our research tremendously because we had to maintain social distancing guidelines. So we could not be in the lab all the time. So we had to, improvise and be more creative, but it also made us more collaborative. And some of the animal experiments that we were planning, we had to actually put them on hold. And some collaborations, between US and Europe, that also got slowed down. So that's how COVID-19 has affected us.
And more specifically, as we all heard, there are, of course, people who continue to struggle with symptoms that seem to be very similar to ME/CFS after they had contracted COVID-19. What are your thoughts about the similarities or differences between COVID-19 and ME/CFS, and how do you think that could be impacted by the topics that we discussed today, and the role of hydrogen sulfide?
Yeah, that's a great question. Actually, the similarity between the COVID-19 long haulers and the ME/CFS patients are strikingly similar. And of course, COVID-19 is caused by a very specific virus, SARS-CoV-2. But ME/CFS is also caused by infection by viruses and other microorganisms. And we are more than 7 months. We have passed well over 7 months after COVID-19, shutdown. And so it is very likely that a lot of the COVID-19 patients will be classified as ME/CFS patients because they go through this chronic fatigue. And emerging research already suggests that the survivors of the COVID-19, they have elevated levels of hydrogen sulfide in their blood. So that suggests something. And based on the results we already have, so we know that H2S metabolism is dysregulated in both COVID-19 as well as ME/CFS. And one good thing which has come out of the COVID-19 is that maybe the funding agencies and other scientists will pay more attention to ME/CFS because it's a real disease. And most people say that it's all in the head when you go to a physician, but it's all in the head because they don't know what's going on in the body. So nobody knows the mechanism, but then hopefully this will open up additional avenues of research, such as redox signaling, which is disrupted in both of these conditions.
Yeah. So specific, you mentioned the metabolic disturbances. There's one question that came in from Cheryl, and the question is, can hydrogen sulfide actually lead to or cause metabolic acidosis?
It could, because it depends on the dose that you use. So now H2S has got a biphasic dose response curve. So at low levels, it's actually beneficial, but at high levels, it can cause all kinds of problems. So we are talking about a window which is optimal, where it will work. But at high doses, yes, it could lead to all kinds of problems, sepsis, etc.
And when you look at that, you mentioned there's a window in which it would be optimal. Can you look at that in blood in real time? There's a question that came in specifically. The question is stating that people at Harvard are right now looking at oxygen level in the blood. But can you actually monitor hydrogen sulfide or other other gases in blood in real time in ME, CFS, and perhaps compared to other conditions?
You can look at the levels of hydrogen sulfide by a method which we call it as sulfidration, because H2S gets scavenged very quickly once it's produced. So it doesn't hang around like a free gas. So instead it exerts various effects. So it causes modification of proteins. So it modifies cysteine residues of proteins, and that process is called sulfidration. So you can definitely look at the levels of sulfidration in patients. You draw the blood and look at how many what's the level of sulfidration, overall sulfidration. And an interesting aspect of sulfidration is that it is decreased during aging also. As we age, the levels of sulfidration come down. And so an interesting experiment to conduct would be to take the blood of patients with mild COVID-19 and those with severe COVID-19 and those with mild ME/CFS and severe ME/CFS. So we are talking about the severity of the disease here. And so when we compare between the two, we need to compare appropriately. But yeah, you can monitor by looking at the post-translational modification.
And to your knowledge, for ME/CFS patients or in other conditions, are there any data on that?
Yes, so there is data on that. So recently in 2019, we published a paper in collaboration with Dr. Filipovich in Germany, where we showed that levels of sulfidration decrease during aging and neurodegeneration. And this is evolutionarily conserved across species, from yeast to C. elegans to Drosophila to humans. And so you can monitor that.
That would obviously be one of the potential next steps to do that.
Yes.
That's great. There's another question from perhaps more from a treatment perspective. Would hyperbaric oxygen reduce hydrogen sulfide and potentially help ME/CFS patients?
Oxygen usually, if you have high levels of oxygen, that will usually oxidize the hydrogen sulfide. But it all depends on what we are going to find in the ME/CFS patient. Are the levels high? Are the levels low? So the treatment will vary accordingly. There are already some drugs, H2S donors, which are on clinical trials for various conditions like hypertension and inflammation. So it all depends on what we find. So at this point, it's too early to say whether that would help or not.
Yeah, well, of course, that remains to be seen, and we're all going to be watching that very eagerly. So I think with Marian, maybe to go back to you. When you see the progress that has been made, from the time of your very first idea to where we are now, what are your reflections, and where would you like to see the next steps? Where do you hope that we go from here?
Well, I hope that there will be more research. There's so much to find, to learn from other areas where hydrogen sulfide is being very carefully looked at. And that focus needs to shift to ME/CFS because I think it holds so many keys. I just hope I hope that NIH in particular will begin to look more carefully at this area. It has changed dramatically over the years. And we now know that it's something we absolutely can't live without. It's something that was previously very toxic, and now we know incredibly beneficial. It's all about getting the right amount. And by the way, your genes affect it, so.
Yeah, certainly, it's gone a long way, and there's more for us to do. And of course, your contribution really has gotten a lot more people interested in this research. So we're obviously grateful for you to provide for providing this spark. Carol, before we close that part, any additional comments and thoughts from you on looking at how bringing people together from different disciplines, different backgrounds? How does that help all of us to continue the search for solutions for ME/CFS? This is the unique role that Solve can play, right?
Yeah, it's understandably difficult to get research scientists in universities and industry to work effectively together. And yet we have found ways to do it. And I think nonprofit organizations like ours, who have, we only have one objective, which is to solve the disease. And so we come at, the attempt to bring people together from a pretty pure scientific perspective on improving the lives of people with suffer. And so, I am proud that Solve ME/CFS initiative has played a role in that, and particularly kudos to our Dr. Sadie Whitaker, who it brings together people both for our registry, where we're now involving certainly we've started here in the United States and now involving folks in Great Britain and Australia and more to come. And so I think it's a strength of hers and has been of our organization to be agnostic about from whence the wisdom comes, and pursuing researchers and helping them find each other, as we have with Bindu and Marian. So I'm proud of that.
Thank you, Carol. And so maybe the last question, Dr. Paul, is what are your expectations when we know more, and what are your plans for the next steps, and when will you be able to share more data with the community?
Yeah, so our next steps is to look at sulfidation and the expression and activity of these enzymes in the Solve ME samples, and also in the cells which you're going to receive from La Trobe University. And I hope very soon, when Johns Hopkins opens up all its facilities, we'll be able to generate more data. We already have some, and I've been doing some analysis on existing data as well. So very soon, I would say we would be able to provide more insights into what's really happening with H2S.
Thank you. That's very encouraging, and of course we will continue to follow that with the hope of more data and potentially new ideas for treatments that may result coming out of that. So we received a few more questions for which we will try to provide more answers in writing. So thank you for those who shared those questions with us today. I wanted to, before we close, it's really an opportunity to thank all the participants today, but really all of you. We do this work with the support and the help of the community, and we're grateful for all of the unwavering support that we receive from you over the years. And especially at this time, which is so difficult for so many of us and around us. So I just wanted to again thank you for your support, and I wouldn't be able to do my job if I didn't ask you to continue to support us. It really starts with you, and it is for you. And please do stay in touch. We are now gearing up towards the end of the year campaign, in which we will continue to rely on your support for making everything that we do continue forward. And at this particular time, I will also mention, perhaps for the first time, that we want to grow. You've heard today about some of the research that is being opened up. We recognize there's much more that we should do and that we can do. We want to expand our research activities, including expanding the registry, as we've mentioned, to capture the experience of people with long COVID symptoms. So we are looking to establish a growth fund, which would really enable us to do more. So we'll talk more about that in the coming months. But I just wanted to mention that we really are very much dependent on this support to continue the research and to continue the advocacy on behalf of the ME/CFS community. So I wanted to thank all of you for participating in the webinar today. Hopefully you've been inspired by Marian's story, and I wanted to thank Dr. Paul and Carol for sharing that webinar with us today.
Thank you.
Thank you.
So thank you, everyone, and we'll see you next time.
Okay.
Thank you so much. Great to
Thank
see you all.
Thank you. Thank you for having
so
us.
much.